A tool with the power to cure all disease or a tool to self-destruct humanity, debates have been sprung up to fight on the morality of the cons and pros of this revolutionary tool. What am I talking about? Of course, I'm talking about CRISPR-Cas9. Good afternoon, everyone. My name is Mustafa Al-Mashhadani, and I'll be speaking to you about one of the most important breakthroughs from modern medicine, CRISPR-Cas9. For starters, CRISPR is naturally found in bacteria. The only thing that we've done is manipulate this natural mechanism into labs. So if we want to understand what CRISPR is, we need to understand the acronym. By breaking it down, we can see that DNA is palindromic, meaning that there are repeat segments, and these repeats will all be identical. Also, we're going to find clustered regulatory interspaced DNA, or spacer DNA, which is going to have variety and is specific to a virus's sequence. And so, let's put all this into perspective. These segments are the identical repeats, and in between these segments, you'll have spacer DNA, which contains variety. And we will also have some CRISPR-associated genes, or Cas genes. They're like many proteins, including nucleases, which I like to think of as scissors. Let's see an example. So let's say this is a bacteria. Cas genes will make Cas nucleases, then a CRISPR RNA is made from spacer DNA and will attach to the Cas nuclease. Let's say a virus attaches and inserts its genetic material. The protein containing a corresponding CRISPR RNA will attach to the viral genetic material and will cut the genetic material, preventing an infection. Therefore, CRISPR is a bacterial immune system. But what if we don't have the specific spacer segments for recognition? Well, CRISPR has a solution to that. So, a virus just inserted an unrecognized DNA sequence. Therefore, Cas genes will produce a class 1 Cas protein. Here, the class 1 Cas protein will bind to the new viral DNA and break it apart, leaving a safe fragment to be incorporated. And so, the class 1 Cas protein will take this cut sequence and insert it into the bacterial genome, so that in the next invasion, the bacteria will recognize and destroy the viral DNA with Cas proteins. Yet we still needed to find a way to incorporate CRISPR in humans. So in 2012, in the labs of Jennifer Doudna and Emmanuel Charpentier, they made the Nobel Prize-winning discovery of Cas9, which looks like this, but we'll stick to an illustration. Discovered in Streptococcus pyogenes, Cas9 works like the Cas proteins we previously saw. Essentially, we'll have CRISPR RNA bind to the Cas9, and this is how it would look like. We'll have two parts to focus on in Cas9. The first is with the CRISPR RNA, complementary to a virus and a spacer DNA, and then we'll have the tracer RNA, which will hold and stabilize the CRISPR RNA in Cas9. Then Cas9 will work like any other Cas protein. Yet unlike Cas proteins, we can modify Cas9 so we can edit human genes. The problem was Cas proteins were too complex to use at the time, so scientists did one simplification to Cas9 for human gene editing. Essentially, the scientists joined the tracer RNA and the CRISPR RNA to form a single guide RNA, making everything else simple. So in gene editing, the first part is the Cas9 protein, and the second is the guide RNA. And we select the sequence of the guide RNA so that we can insert the gene wherever we want, preventing any damage to existing coding DNA. With these two parts, Cas9 will approach the selected DNA based on the available guide RNA. The DNA will feed into the Cas9 like this, and the DNA strand will be cut in a selected area to prevent damage. Then a genetically modified DNA strand is inserted into a cell and will attach to the cut DNA strand, now being incorporated into the cell's genome. The inserted DNA will be used to make all sorts of target proteins. For example, we can repair pancreatic cells to cure people with diabetes type 1 and restore insulin production, or we can treat people with sickle cell anemia, which causes non-functional blood cells, like with the case of Victoria Gray in 2019, who was successfully treated with CRISPR-Cas9. We can also use CRISPR-Cas9 for COVID-19. First, Jennifer Doudna made another invention last year, which is a fast and effective COVID-19 diagnostic tool using CRISPR, which is much superior to current PCR tests. What you need to know is that a modified Cas protein will be inserted into a sample containing a corresponding guide RNA to the virus. If the guide RNA attaches to the viral RNA, it will produce fluorescence that can be seen after 20 minutes with just a smartphone camera. That's how simple it is. Another usage is with artificial intelligence. Right now, there's no medical cure to COVID-19, but CRISPR and AI are about to change that. And so Stanford University has been able to genetically engineer a Cas RNA with the help of AI that corresponds to COVID-19 RNA. The Cas proteins will bind to the viral material and cut it to deactivate the virus. We have already seen multiple COVID-19 variations as well. As variations will have different sequences, they require specific guide RNAs. Artificial intelligence allows us to quickly sequence new variants of COVID-19, so we can quickly produce guide RNA, so Cas9 can recognize viral RNA. Of course, the issue of gene editing brings a lot of ethical concern. The first one is about the choice of body. Would there be peer pressure or discrimination against people with or without gene editing? Another point is about designer babies. Are we allowed to change our children's genetic characteristics for non medical purposes before they even have a say? And finally, will healthcare providers force people to undergo genetic change to be eligible for healthcare plans? These are all questions that we must answer now with more CRISPR awareness as we are running out of time. Yet we know that CRISPR is the future, as it can cure some of the most deadly diseases like diabetes, cancer, or future pandemics. Thank you for all for attending my symposium, and I really hope that you learned more about CRISPR, and I hope that this was helpful to you.
Thank you, Mustafa, for your insightful and productive presentation. The judges may now vote. Everyone, please welcome Razan from Al Mawaqif Garhoud, who will be our last panelist discussing artificial intelligence and COVID 19.